is my pleasure to introduce our speaker today. Cyril Pommier is a bioinformatician with a background in molecular biology and computer science. He's been working since 2007 on plant phenotypic data management in global data federations, trying to build bridges between field experimentation, molecular biology, and computer science. He is a co-leader of the ELIXIR plant community and the ELIXIR biodiversity, food security, and pathogens area. He is also a co-leader of the data work package Phenomics, which is the national infrastructure for plant phenotyping in France. And yeah, I will turn it over to Cyril, and thank you very much for joining us.
Thank you, Peter, for the introduction, and thank you, everybody, for the invitation. I'm going to share my screen. Here it is. So, of course, I'm going to share the PDF of the presentation that I'm going to give. It is mostly what I've been presenting in PAG, but with the fusion of two of the presentation I've made, both the computer demo and this workshop presentation around fair plant data phenomics data management, the current status, the tools, the guidelines, the way to handle this challenge, both in ELIXIR and emphasis European infrastructures. So, as an overview, I'm going to try to guide this presentation along the data life cycle for phenomics data, but which can, of course, be applied to a lot of different type of research data. The first step is about planning, then collecting the data, processing it, analyzing it, preserving it, sharing it, to then finally enable the reuse of the data. The important thing, of course, is that there are several tools already available, platform information system, which most of the time are rather specific of communities or experimental platforms. There are also portable devices, sensors, which are also very project or community specific, scripts, workflow, and finally data repositories, which can be generic, such as Dataverse, Recherche.gov, which is a Dataverse, Zenodo, or way more specific, such as the ENBL, EBI equivalent of the NCBI in Europe, and the FIS information system for phenomic data, which you had a presentation about back in October. And since there are many data repositories and platform information systems, there is a need for data portals to be able to search over all of those. So since there are a lot of tools, a lot of solutions available everywhere, we need something to glue, to link all of those solutions, and those are twofold. First, guidelines, community practices, and I'm going to elaborate on what is being done in the RTM kit, and also global data standards with MyAPI, the breeding API, which are somehow two faces of the same coin, and the MCPD for plant biological material, plant genetic resource identification, as well as the crop ontology for traits. So the first thing I would like to sketch the scope of the tools we have been working on, which is mostly around phenotype to genotype integration and management, with the observation, of course, that phenomics are dispersed. There is no central repositories like NCBI or EBI, nor any plan, as far as I know, to build one. The data are very heterogeneous and are more and more standardized. On the other hand, we've got omics, genomics, genetics data, which are mostly centralized, but some are managed in a decentralized way. They are mostly homogeneous. A genomic feature is the same in a cattle and in any plant species, with some particularities for plants, of course, but the metadata can be very different. There is different way to identify a sample, an individual, a genetic resource between wheat, forestry, and of course animals. And finally, there is a need also to manage environment data, but mostly in linkage integration with Phenomics data. And we need to link all of those, integrate them to be able to run analysis for plant breeding, so genetic variations by trait studies, or climate change studies, genotype by environment. So to build plant data standards to answer to those challenges, to those needs, several European research infrastructures have joined forces. First, the ELIXIR, European Infrastructure for Life Science Data, with many countries involved. There are 5 platforms, compute tools, training, interoperability, and data. And a lot of communities currently, there are way more communities than that, something like 30 of them. Those which are of interest for today's talk are, of course, mainly plant science, which I'm co-leading with Sebastian Bayer and Christina Gruden. And although there are strong connection with, well, connection to be elaborated with biodiversity, food and nutrition, as well as pathogen. And we've got almost 10 years of collaboration between this plant science community and the emphasis infrastructure for plant phenotyping that gathers although many European countries, which has a data and computational services work package. And another thing which is, in my experience, important to precise is what we are talking about when we talk about phenotype, because in fact, this word encompasses a lot of semantic diversity. And this is due to the complexity of the data lifecycle of phenomics, plant phenomics. Indeed, we've got a first step here in green, which is about data acquisition with raw data measure, observation of phenotype on environment variables. Here, this green square symbolizes either a greenhouse or a field. And with this, we can have time series table of measurement with rather complex experimental design. We can also have rather complex series of images. And those data are then derived, reduced to prepare them for analysis, since most of the time they are too complex to be directly analyzed. And those derived data, those indicators, allow, for instance, to have a single value for a given variety, Soissons here, under high nitrogen cultural practice. In that example, it is a mean, but it can be very more complex. But even those derived data can be rather complex with time series that can be rather dense. And from those derived data indicators, we can then extract the knowledge, such as variety charger, its resistance to fusarios in their intensive cultural practice. The thing to keep in mind is that the data acquisition, we are mostly dealing with variables, with raw measures, phenotype, and environment. We have to deal with data cleaning, with traceability, reproducibility, and provenance. For derived data, we are not dealing with indicators that integrate many variables. As a consequence, since those indicators are built for each scientific question, each study they are built for, we observe that one experimental data set can lead to many derived data sets. Therefore, it is absolutely paramount to be able to keep on the long term the raw data available and correctly described. And to describe them correctly, of course, we need several data standards. The first set of data standards is semantics. It's about description of the data, about building controlled vocabularies, list of term names, as well as their definitions, upon which the biologists have agreed within each community. Those controlled vocabulary can be made a little bit more complex through their transformation into ontologies, by adding semantic links between the different terms rather than a flat list. The important thing to keep in mind is that those standards, to remain efficient and to remain widely adopted and used, must be biologist driven. They must be built and maintained by the biologist. Then you've got structured data standards, which allow to format and organize the data, and more important, allows to build explicit data models to organize those data and to put different concepts in perspective from one to the other. They can be very simple, CSV here, a data standard, or more complex with VSCF or GFF for genomic data, or MyAPI for phenomic data. And in our experience, to build successful structured standards, we need to have both biologists and computer scientists around the table. And finally, when you've got to deal with more technical challenge of data interoperability, of integration, of connection of tools and systems together, we need more technical data standards such as GA for GH for genomics, or the breeding API for breeding, so phenomics, germplasms, and genotyping data. And this is definitely the realm of computer scientists. And in the middle of that, we need a glue, something to link all of those, to identify correctly the different concepts, the different resources, data resources we are dealing with. So with persistent unique identifiers, DOI or URI, which provide an ambiguous namespace at the scale of the World Wide Web to identify genes, accessions, so plant genetic resources, traits, and so on. So I will not detail all of those three standards because first, I'm assuming for BRAPI you had or will have some presentation. Crop ontology should be the case too. I really want just to focus today on the structure standard, MyAPI, which stands for Minimal Information About Plant Phenotyping Experiments. So of course, we've got a website, www.myapi.org. This standard addresses the needs of crops and woody plants mainly, but it can be, of course, very well adapted to model plants such as Arabidopsis. It can handle single experiment or multi-local, multi-year networks of fields or greenhouses. It has been built by many stakeholders, ELIXIR and emphasis, of course, but also biodiversity of the CGIAR, in other words, crop ontology, who are part of the steering committee, or the North American Plant Phenomic Network. It is an open community with requests for comment, GitHub feature requests, mailing list, meeting, work groups, etc. Just to get back on the history of MyAPI, there has been a first version, which was an initial checklist in 2015, a big update with the addition of the formal model, data model in 2019. And there is currently a request for comment for a new version, which has only few additions, few additional fields, which is, in my opinion, a very good sign because it shows that the standard is stable and is now totally in its adoption training phase. We are more dealing with the question of making it implemented in different systems rather than developing it itself. So the RFC, by the way, is still totally open. So if you want to contribute, you are very much welcome. So MyAPI is a minimum information for biological and biomedical investigations. So this means this is something that is mostly say biologist friendly, with clear definitions and examples, with Excel templates, because it's very straightforward to explain things using those tabular data, and with a lot of focus on trainings. But it is, as I said, also computer scientist friendly with data models, implementations, and formalization. It is a minimal and sufficient list of metadata with the objective of the experiment, the person who contributed, what were the experimental procedures, what was the biological material experimented. The important thing also to keep in mind is that it's only a specification that needs to get implemented in different systems. First, mainly to formalize things, there is an ontology, an all implementation available on GitHub on the Agro portal that we can see here. It is a representation of the data model with formal concept and constraints. There are also file archives implementation with historically ISATAB, which is somehow a zip archive that can handle both data and metadata. There is also some work initiated on EROCrate, which stands for Research Object Crate, that somehow holds the same promise as ISATAB to have a self-sufficient archive to exchange the data and load it or unload it in any tools or system. And of course, there is the Excel template that can be rather well used. For the web services implementation, the breeding API is the MyAPI implementation. As you know, it's an international collaboration with information exchange, main target breeding, and with the coordination support from the Excellence in Breeding platform. And finally, there are several data repositories, databases that implement MyAPI. So de facto, any BRAPI compliant database is also MyAPI compliant. It's the case for GNPIS in France, FIRS in France and beyond, PIPA in Emphasis, as well as generic data repositories such as Dataverses, Zenodo, or EDAL. To give a very quick idea of what exactly MyAPI is dealing with, we'll give an overview. You will have more details through the MyAPI primers on myapi.org or on the paper from the new phytologist 5 years ago now. So MyAPI is the specification table that you can see here. You've got 11 sections with investigation, study, experimental factors, observation unit, sample, observed variables, and so on. Sorry, I was a little bit too quick. And within each of the section, you've got a list of fields, such as here's the variable ID or name, as well as their definitions. Few examples. Format and cardinality constraints. And on top of that list, you've got the model itself that allows you to bring together all those fields, all those sections and concepts, and shows that an investigation holds several studies, which hold several observed variable and several biological material, etc. So MyAPI is based on the ISA backbone. ISA standing for Investigation Study Assay. It is, as you probably know, a standard used mainly for omic data, but which has been successfully extended to Phenomics. So the investigation is a world data set. So a collection of studies of experimentation. Each experimentation or study is in one location for 1 to several years. 1 to several years, of course, because if you are dealing with perennial plants, such as trees or grapes, you will run measures and observation year after year. And finally, you've got the assays, which are the trait or indices or variables that are being observed and measured. And they are made of the triplet trait method scale unit from the crop ontology, plus the addition of a level where it has been observed, a level being the plant, the microplot, the block, the trial, and so on. And all of those are mostly metadata, description of the data here. And therefore, you are able then to store alongside those metadata multiple data files corresponding to your actual measurement, observation, or computed data. There are a couple of sections in MyAPI which are of special importance. The first one is, of course, the biological material. It's a plant material identification and description. It is based on the multi-crop passport descriptor. With the building of MyAPI, we didn't want it to reinvent the wheel, but rather to import existing established data standards and link them together. So the multi-crop passport descriptor used in the different gene banks provide us with different levels of identification of the material. The first one is a material source. It's the plant genetic resource accession. But it can be also the cultivar or the variety, the commercial one sold by breeder. It can be also the region of provenance, which is important for forestry, or of course, laboratory material, laboratory cross, or any scientific material built for a given purpose. The important thing is that it is always an identification system based on a triplet. The gene bank, the laboratories, the breeder, well, a namespace for unicity. The species, in case you've got several species within a given gene bank or breeder. And finally, the accession number. Also note that the MCPD provides the possibility to put together this triplet in a single DOI, which is super comfortable, but it is absolutely not generality a system that is widely and uniformly adopted. Then you've got the biological material used in the study. So this is your physical plants, the seedlot, the cuttings. So once again, it is identified by a laboratory plus internal accession number, sorry. And that can be summarized as a single URI. Finally, of course, you've got plant samples used in the study, such as detached leaf, which can be identified for interoperability using biosample ID provided by the biosamples database. For the observed variable, so there is a plant phenomics specific ontology model. While for gene ontology, sequence ontology, etc., we are dealing with atomic model. For phenomic variable, we need to rely on a triplet, once again, with a trait, a method, and a unit. The trait can be, for instance, the plant height, such as in this example on maize. The method can be the total height, first tassel branch, etc. And there are really a lot of different measurement methods. And furthermore, they can be community or project specific, which provide a lot of heterogeneity, complexity, and diversity of the number of methods. It can be also rather exotic things, such as here, the highest pixel corresponding to a plant when dealing with high throughput phenotyping method. And finally, you've got the corresponding units, international system, or very specific such as pixel. So each variable is made by the combination of one trait with one of those methods and one of those units. And when you change either the unit or the method, you are creating a new variable. As a consequence, it is very difficult to maintain a reference list of variables that is suitable for any community, any project. This is why beyond the availability of the crop ontology reference repository of ontologies, MyAP offers the possibility to store any type of variable within each dataset. So there are a lot of important sections. I'm not going to detail them here. I will also note that the data file content itself is totally free. MyAP doesn't constrain the data file structure. It can be near infrared spectrum, images, image archives reference, etc. But so far, I've seen mostly tabular files, such as this one. As a consequence, some people requested MyAP to build a non-mandatory example of what can be the data file acceptable for MyAP. So there are some perspectives which are mostly listed and proposed in the different GitHub issues and are discussed within the steering committee. There is this question of a recommended data file format that I just mentioned. We are also working on extending standard interoperability toward ICASA that is being used for agronomic model exchange. It is under study and with personally, I think the idea to import events from ICASA into MyAP and to bring biological material to ICASA. But we are still in the initial phase of that work. So the next thing I'd like to underline are the guidelines, and especially the RDM kit that allows to share guidelines, data management guidelines within communities. RDM kit is the best practice and guidelines for fair data management. RDM standing for Research Data Management. It's a Wikipedia-like knowledge-based website, free and open. It describes how to manage research outputs according to fair principles. And it's not a central repository of guidelines. It's a portal to other online resources used by RDM professionals and researchers. Note that this website, this Wikipedia-like knowledge base, is recommended is the Horizon Europe program guide. Horizon Europe being a set of calls, grants. Yes? So RDM kits, there are nearly 200 contributors, a lot of tools and resources described there. Contributors are experts in research data management or in scientific domain in ELIXIR infrastructure and beyond. It is managed by an editorial board with ELIXIR members, but as well as people from the NIH. The content is created by members of the ELIXIR RDM community. The organization of the RDM kit is what makes it really straightforward and easy to use for biologists, because you've got domain pages such as the plant science page, for instance, which is my center of interest. That gives you an overview on the data management planning, the plant biological material handling, phenotyping, genotyping data, etc. As well as, so you've got here the link to this plant page, as well as links to different tools assembly. Tool assembly are a way to make different systems, standards work together for a given purpose. For instance, you've got one plant phenomics and one plant genomics tool assembly available in the RDM kit. Each of those tool assembly gives the possibility to link from the RDM kit to the recipe detailed repositories, such as, for instance, a FAIR cookbook in that example. The plant genomic assembly is a step-by-step procedure to publish data at ABI. It ensures interoperability from phenotype to genotype. It is available at this website. Also, you've got the plant phenomic assembly, which guides users in publishing data in generic data repositories such as Dataverse, Zenodo, or IDAO. So I did a computer demo on that at PAG. And just to go quickly into the details of how you can do this, the recipe in the FAIR cookbook from this tool assembly show you how to create minimal metadata, the minimal data set within those generic data repositories, such as here, so the Dataverse, but with no limitation. Mainly is the fact that the metadata are too broad, with year, data type, author, organism, which is not controlled by any kind of vocabulary, only keyword and subject, and that's all. We are lacking all the plant phenomics specific metadata, such as biological material, trait, experiments, location. So we need, of course, in those data repositories, which have a mandate and a dedicated fund to be still available in 10 years, and which are therefore very interesting. So we need dedicated metadata scheme added to those generic data repositories. So the approach we have put forward is to add additional metadata as companion files, which is the most convenient way. So by using, of course, the MyAPI data standard. So the way to store this in Dataverse is to use templates coming from MyAPI, biological material, observed variable, adding those lists of fields to correctly identify your material, and simply submit them alongside the data files. So there is here an example of such a well-defined dataset on a maze multi-site dataset, where you can see you have this observed variable tab with this little annotation that shows this is a MyAPI standards file. Same for the study. You see that in each of those files, you've got the list of variable ID, variable name, accession number, etc., conforming to MyAPI, and the geographic location, latitude and longitude for each 20 experiments. And you've got, of course, a biological material tab, once again, identified by this annotation that will not only allow biologists to identify metadata files, but also allow automatic indexation by data portals. Finally, we are also pushing forward the building of human-friendly provenance to show the relationship between all the different files. The next step after that is how to enable release interoperability between the different data repositories, and we have published in them. So while MyAPI, of course, is very well suited for phenotyping databases, either natively or as breeding API compliant databases, we have worked with within ELIXIR with EBI to enable the same data standard within the biosample database at EBI, which enable to identify the samples for sequence data or VCF data using the same set of fields, hence enabling very easy data linking between the two data types. To first, of course, being able to build integrated dataset, or at least to find related dataset through community data discovery portals such as FEDER. So FEDER is one of the data portals that is being proposed in Europe. Florilège is built in France and especially for plant genetic resources. FEDER is a global plant research data discovery portal that indexes sources conforming to MyAPI, both from Phenomics and genetics or genomics, as well as generic data repositories such as Dataverse or the WIT-IS WIT initiative databases. It indexes currently 33 databases all over the world. It provides a very simple full-text search with ontology-based synonyms, here entering peuplier in French, generate hits on populus and poplar. We've got also dedicated filter, which are mostly provided by their compatibility with MyAPI, with data types, taxonomy, genetic resources, crop ontology, gene ontology, and so on. And the community management, the ease of joining this federation is something absolutely instrumental to make it success. And so we are making it very easy to join this federation. So I will not go into details of the demonstration, of course, just to show you that each data resource can be pre-visualized within FEDER, which can be useful for data sets that don't have a dedicated database, such as here, this phenotyping study that shows in detail the list of genotypes, so the germplasm used, as well as the variables studied in this study. So to finish, the take-home message is that alongside this data lifecycle, there are a lot of existing solutions and tools that can be either very community specific, especially around the first steps, around platform information system or portable devices, sensor, measurement means, etc. But there are others that can be more easily shared. That the community management is absolutely critical to ease the adoption and the sustainability of those tools on the long term. Hence, the importance of the RDN kit. Before the RDN kit was created 4 years ago, there was the equivalent built within the WIT-IS of the WIT initiative, and it was rather well welcomed. So it is something important. Interoperability is also critical. It is built on very concrete use cases, scientific question, and use global data standards. There are global partnerships that exist, and RDNKit is probably the entry point that allows you, after this talk, to get more details on all those tools system available. And I'm not going to list everybody, but everything that I've shown you, of course, has been built by a lot of people. And I'm done.
Excellent. Thank you, Cyril. We'll open it up for questions, either questions in the chat or you can unmute yourself and ask.
Thank you for the presentation. Really enjoyed it, and this is I learned a lot from these activities, and especially happening in Europe. My question is that these days a lot of phenotyping data is coming from images, drones, or you still show an example of indoor phenotyping facilities. But what is the status of? Because it looks like once we extract some phenotypic information from images, that goes into this standard and that being going to the database. But is there any activities that happening around how to incorporate those images to be part of this standard as well?
So I'm not sure I get exactly your question. Let me rephrase and please correct me if
Sure.
I did not understood well.
Mm-hmm.
So the challenge you are pointing is how to deal with first vast quantity of images that are being produced, how to correctly document how tabular data have been extracted from those images, and how to manage efficiently those images and link the tabular data to the images. Is this your
So yeah, I
question?
think sorry, probably my question was not clear. I mean, probably that is because my background is not coming from this area. But for example, let's say going back to the indoor phenotyping the facility the case where you take this in RGB images, you measure the canopy height from those, and
Mhm.
looks like those measured canopy height is going to the database, but not necessarily the original images is being tracked or imported into the database, so that, for example, some other people they have a new way of measuring same information in different way or something like this. So I like to see how this the original data that gives that specific measurement can be uh, being managed and shared and in this framework. So I know this is a probably very open-ended question, but I just wonder if there's any been discussions or activities happening around.
So, in my understanding, this is a question of provenance, of traceability, of documentation, of how you extracted a trait from an image or set of images, and how you link back to those set of images to be able to run a new set of analysis on them. There has been a lot of discussion for at least 5 years or something like that, or even more. 5 years for what I know, but I'm not a specialist of that question.
Mhm.
There is ontologies such as the Prov ontologies that allow to formalize that. They are not super user friendly, and it might be a reason why it has been difficult to adopt these Prov ontologies so far.
Okay.
Anyway, there is a need to be able, within those standards, to store those information, and also within the tools. And I'm thinking about fields that has a section with provenance that allows to link from a tabular data to the original images with all the information of the software used to do the actual analysis. But very honestly, this is not well formalized today. I've got a series of leads, but this is me in my own mind thinking about it.
Thank you very much.
Peter, you're welcome. Peter, this question might have been discussed during the different RAPI hackathon. I don't recall this very well. Do you want to elaborate on that, or?
Yeah, and Jinhan have spoken in the past as well, but the I think provenance is a important aspect, as well as just being able to have the metadata for the images themselves. What they what they were taken of, what they represent, what sort of sensor camera was used, that sort of thing. So yeah, definitely within the BRAPI community, we've begun looking at standards for image metadata in particular, and yeah, I welcome more input on that. There's also an effort. Seth Murray of Texas A&M is trying to build a community around drone data specifically, but sort of generally high throughput phenotyping to try and discuss and land on some of those standards. So I think, yeah, I think the discussion is ongoing everywhere.
Yeah, yeah. And there are also a project being submitted now about a connection between Phenomics and imaging data through the Euro bioimaging infrastructure. And I think they've got standards and tools to manage mostly microscopy images, but at a rather big scale. So I'm hoping this would help, but we will see if the project succeeds.
I mean, it's good to hear that there is no such a solution out there already, because those are the areas that I'm trying to work on, and I'm glad to hear that there is some needs out there. We need to have such a standard needed for this area. I think that those indoor phenotyping images is good, but another huge monster in this room is that Peter already mentioned drone data that large, tens of gigabyte for a single file, and how we can actually effectively manage and share and visualize all those kind of stuff. So I'll be working with Peter and Jennifer in this room. We started a new working group called the Phenotyping Data Standard Management. So those are some things that we'll be discussing. Cheryl, probably you are going to hear from us later about the finding that we
With
got.
pleasure.
Yep.
I have a quick question from Jennifer. Are these resources limited to institutions within the EU and emphasis?
No, absolutely not. There are definitely global standards. For my EPI, there has been a contribution from North American Phenomic networks. Those tools have been also adopted by DDBG in Japan, and we are working also with Narrow. FEDER is indexing databases all over the world. So no, this is way beyond Europe.
All right. Any other questions or comments or anything for Cyril?
Thanks, Tanner.
All right. I think that's a good place to end it. Yeah. So thank you everyone for joining us, and thank you, Cyril, for presenting.